morning. Thank you. I would like to share with you a part of my research project and titled "In Vivo Characterization of Ileal Microbiome in Healthy Cattle Using Metagenomics." It's the importance of the ratio between firmicutes and bacteria deaths in the GI tract. It's well described in the literature, and its importance. It's very important into the overall bacterial and host metabolic homeostasis. In ruminants, there are also several studies out there that look into the rumen microbiome and its influence on different physiological parameters in the dairy cow, per example. But there's not a lot of done in the intestinal microbiome in vivo in the literature. In other species, in mice, for example, it's really interesting. They have been able to show that this ratio between the two bacterial phyla affects and is affected by obesity, and how also it plays an important role on how the host harvests energy from the diet. A group out of Canada published an article back in 2013 that they look into the impact of the antimicrobial growth promoters and in the gut microbiota. And those samples were collected at slaughter, and they were indeed able to show a huge variation between different sections of the GI tract, but mostly importantly, they showed no impact of the usage of growth promoters into the differences that would may or may not cause into the microbiome present. A lot of these works, a lot of the work published has been done in post-mortem, either at slaughterhouse or shortly after euthanasia. So we think it's really important to know what is the picture in vivo, what's happening while the animal is within its normal husbandry. So our objective was to describe the commensal microbiome in the lumen and at the epimural surface or the mucosa of the ileum in cattle in vivo, as we hypothesized that there were differences between those two locations when we determined that. What we have done is we had 6 dairy crossbred adult steers. They were about between 14 and 16 months of age. We surgically fitted these ileal T-shaped cannulas and to allow the sample collection and so forth, so on. There were about 3 to 4 month recovery period between surgery and the actual research project, when the research project took place. And the reason for that is we wanted to make sure that the animals were recovered well from surgery, and we did use antibiotics and anti-inflammatories at the surgery day. So we want to make sure that those medications were gone from the system by the time that we started. And those animals were in a strict husbandry situation where we fed them the same amount of feed, and they were kept in the same area. Water consumption, everything, we tried to make sure that there was not a variation that could impact our results greatly. In order to reach as much as variation as possible, we collect samples over a month periods of time, and each animal went through the collection 3 times. This allows us to come up with 18 samples from the lumen of the ileum and 18 samples of a biopsy of the mucosa of the ileum. And for the luminal samples, we used a 20 centimeter Foley urinary catheter that we fed through the cannula and collect our sample that way. And in order to collect the biopsy, we used a flexible video endoscope, and you can see on the picture on the right side of the screen. We used so we fed the endoscope through the cannula, and went in an average about 50 to 60 centimeters into the ileum to collect our biopsy sample. The DNA was extracted at our lab, and we use a commercially available kit to process those samples. And once that was done, we submitted them to a private laboratory where microbial 16 sRNA ion tag and community metagenomic sequencing using the Ion Torrent PGM analysis was performed. So there was about a total of 1.7 million reads that we found. And pretty much we looked at, we look at the phylum level. So we picked the top 7 reads at the phylum level and compared them between both locations, the lumen and the mucosa. We have not done any statistics analysis yet. There is as y'all probably can imagine, there is a huge data set sitting on our computer right now that we need to continue looking. But our goal here was to determine what was there in vivo. So a lot of the data is still being worked out, and we're trying to get that done using the appropriate softwares to analyze microbiome data. But the differences were pretty interesting to us. Once we compare the two areas, and on the lumen, there's a lot of firmicutes, bacteria showed there, and interestingly enough, the bacteria phylum, the bacteria associated with that phylum has been shown to induce obesity in mice once in a heavy fat diets. So that makes us think that that it is associated with perhaps the type of diet that these animals were getting. But nonetheless, interesting results we thought. As described in the literature, the firmicutes and bacteroidetes, they composed over 80% of the microbiome in the gut. So I want to show you that, but something really interesting happened here, and the next 2 slides I'll show you, we're going to look at them separately, bacteroidetes first and then the firmicutes. The duodenum on the bottom left, that is a data from, it's unpublished data from our studies, and here we were able to observe that there's a really good consistency among animals, among the calves. Everything that's in blue is the lumen, and in orange is the mucosa. So in the lumen, the bacteroidetes were the predominant phylum there, but when we move to the ileum, it is not. So pretty much those results kind of shifted on us, and that was an interesting finding. And the same is true for the firmicutes. The variation between the animals is expected. However, it's pretty consistent when we look them individually. But the results also flipped if we compare to the data that we gathered related to the duodenum and now what we have with the ileum. There are several discussions in human medicine about changes in the gut microbiome and its effects on patients with inflammatory bowel disease. And we know that while imbalances in the duodenum and the fecal microbiome community have been associated with mucosal inflammation. Very little is known about alterations in the mucosa bacteria associated with in patients with inflammatory bowel disease, and when we involve the ileum and the colon. So our goal was to try to determine that in cattle. In the dairy cow, the ratio between the firmicutes and bacteria deaths has shown to affect milk fat production. And a little while back when I mentioned that the ratio of firmicutes were higher in the duodenum, and it flipped on us, and now it's slightly lower in the ileum. A paper published in 2010, they suggested that the concentration of these gram-positive bacteria associated with the mucosa at the mucosa level was the reason for that is those bacteria were taking advantage of the host oxygen. So we speculate that in this case, the reason that it flipped was it had to do with the function of those organs. So in other words, the duodenum, as we know, it's responsible for absorption of iron, as well as there are a lot of the digestion is still a lot being completed at the duodenum level, where in the ileum it has just a function of absorbing nutrients, B12, vitamin B12, and bile salts mostly. So because of that shift in function, perhaps the bacterial population also adjusts to that to make sure that that it's done properly. So it's really interesting finding, and we're still trying to understand all the reasons. But limited research on GI microbiome in cattle in vivo that we could find. We believe that it's a great importance for several reasons, but the main one that's close to my heart, we believe that especially when it comes to antimicrobial usage in food-producing animals, we want to understand the role of those bacteria, and if treating animals do indeed affect those changes. So there's a lot still to be done, and we're looking to the other levels, and obviously trying to determine statistically if there is anything there. As we move, following these studies, this specific study, what we did was we went ahead and inject the animals with a commonly used macrolide antimicrobial in the U.S. For respiratory disease, and we pretty much repeated the same materials and methods, and we analyzed that data as well. So that is being presented by my colleague and co-author later on today. Any questions? Thank you.